and it is a real pleasure actually to talk to the rest of you. Now you actually know what I've been doing all these years. So when did I start? Well, I started in 1998, and some of the younger ones are saying, oh, that was a millennium ago. Oh, yes, too true. It really was the last millennium. How scary is that? So what was happening at the time? So a young chap, Harry, went to school to Eton with his daddy. This is Tony Blair signing the Good Friday Agreement. A little company was founded, very small. Anyone heard of it? It's called Google. And then we had a president be impeached for lying about, absolutely assuredly telling everybody he had had no sexual relations with Monica Lewinsky. And then about 3 months later, he admitted to having sexual relations with Monica Lewinsky, but he managed to get out of this. So what was the state of play with regards to osteoarthritis? So I came to the Kennedy Institute as a rheumatology registrar, wanted to do a period of research, as Fiona said, decided to join Jerry Sacklett-Vala's group, who was at the Kennedy at the time. And we really knew very little about osteoarthritis, and it really depended on who you asked. So you could say that at the time, the best developed science was pathology, because people had been looking into people's joints for a number of decades and centuries, cutting up bits of tissue and showing that actually this is a disease of degeneration of the articular cartilage. There is new bone formation, we call these osteophytes, and you see thickening of the thickening of the synovium or hypertrophy of the synovium. There never was very much evidence of frank infiltration of immune cells in this disease. This was never regarded as an immune-mediated arthritis. So you can see that if you look at human joints, this is a finger joint. You can see very similar things if you look in a mouse joint. All animals of the animal kingdom will develop osteoarthritis, and most of you in this room will develop osteoarthritis if you don't already have it. So pay attention. That's what the pathologists would say. If you spoke to an orthopedic surgeon back then, and I hate to say, but many orthopedic surgeons would still say the same thing today, is that osteoarthritis is a simple wearing down of the articular cartilage. It's like a rubber tire, Tonia. Don't worry about it. You just have to change it from time to time. We happen to believe that this tissue is alive, the cartilage, and therefore it's doing more than just rubbing down. The rheumatologists haven't been very much more hopeful than that either, both back then and now. This is the disease that they exclude. They go, oh, thank goodness, it's not a more exciting disease like inflammatory arthritis. We'll send you back to the GP. So this is a diagnosis of exclusion and has always been regarded as the boring, unfashionable disease in rheumatology. And then, of course, if you speak to the patients, they'll give you a completely different answer. They'll say, this is the painful, disabling joint disease that limits my day-to-day activities. Okay, so there were a number of things that we knew to be true in osteoarthritis back in 1998. The other scientific discipline that was very well described was epidemiology. We knew about populations of patients, and we knew how osteoarthritis developed within those populations. So we knew that this was a mechanically driven disease. I would argue this is the obligatory risk factor in the development of disease. We also knew that there are a whole host of other OA risk factors that enhanced that risk. So for instance, if you had obesity, if you were older, if you were female rather than male, you had an increased risk of developing osteoarthritis. And then from monozygotic and dizygotic twin studies, we also knew that osteoarthritis was heritable. And that is estimated to be around 60% that you could account for by genetic factors. But here are the things that we didn't know. We didn't know what the molecular pathways were that initiated and propagated the disease, or why they were being initiated. We didn't know where pain comes from within the osteoarthritic joint. There are a number of structures within the joint that are innervated, the articular cartilage being the one tissue that isn't innervated, and everyone said this is definitely not where pain comes from in OA. And we also didn't know this other important question, whether it was several diseases all converging on a common pathology at the end, or whether actually this is one disease with some slightly different flavors. And I'm going to try and address all of those today. So I'm going to start with those mechanical risk factors, because these are really absolutely critical for the direction in which my research first went. So we know that if you put too much load through a joint, and that can be because you are overweight, it can be because you have malalignment of your joints, so you're putting more compression on one side than the other, or because you're doing things with your joints that put them under excessive mechanical stress. This will increase your risk of developing osteoarthritis. But also think about the loss of those protective mechanisms within the joint. And that's very easy to understand when we think about the ligaments, the menisci. These are the things that stabilize your joint and prevent it slipping around when you do normal daily activity. So if you have damage to any of those, even if you're a young person, you're at substantial risk of developing osteoarthritis. But also, as we get older, we lose muscle support across the joint, and this is also a really important risk factor for increasing shear stress in the joint as we're using the joint. So that is why when we observed very early on in my scientific career that cartilage was exquisitely mechanosensitive, I thought this has got to be an interesting thing to study. And particularly because when you look at the articular cartilage, it's a really, really unusual tissue. This is a tissue that has really small number of cells. Those cells are surrounded by dense extracellular matrix. That's what's stained purple in this slide. That's the collagens and the proteoglycans in the tissue. The cellular component is only 5 to 10% of the volume of the tissue. The rest is all extracellular matrix. This is a tissue that has no blood vessels. It has no endothelium to damage. It has no ability to activate platelets and clotting within the tissue. It doesn't recruit leukocytes. There's no resident macrophages. So how on earth is it that you take a simple cut of your tissue, you cut it with a scalpel out of the joint into serum-free medium, and you see activation of a whole host of intracellular signaling pathways? And here I'm showing the inflammatory signaling pathways. These are activated within 30 seconds of that injury. This is exactly as strong an activation as you'd see with IL-1 or tumor necrosis factor. And the consequence of this signaling is that you see upregulation of a number of injury response genes. Some of these are inflammatory, some of them are non-inflammatory or possibly pro-regenerative. And over the years, we've been able to show that this very crude in vitro injury is relevant to the in vivo system. We can show that the same injury response genes are regulated in the articular cartilage in the mouse after we destabilize the joint. And we can also show that those same genes are expressed as proteins, and you can measure them in the synovial fluid of patients who've had an acute destabilizing injury of the joint. And so what I started doing in my PhD, and I never really stopped, I'm sorry to be so boring, is to then try to work out how this tissue, which after all is receiving all of the mechanical brunt of this increased stress or increased load, how is this tissue responding to mechanical stress? And we've defined two broad ways in which it does so. The first is by activating what we've called mechanoflammation. That was really to satisfy Fiona. I had to put inflammation in there somewhere, or immune related. It's not immune related. It's part of the innate immune response to tissue injury. This seems to be predominantly activated by that shear stress. So imagine that very unstable joint, and it's sufficient to trigger very proximal upstream kinases such as TAC1, and then further downstream pathways like the inflammatory MAP kinases, NF-kappa B. It switches on those inflammatory response genes, and some of these are critical for matrix degradation because you see a regulation of the metalloproteinases, the aggreganases in the tissue, but also, and I'll come back to this at the very end of my talk, it switches on nerve growth factor, which is arguably the most important driver of pain in osteoarthritis. And remember, this is coming from the cartilage, not elsewhere. Now, conversely, if you squeeze the tissue, if you compress the tissue, then you see a different response. You see release of these growth factors. These growth factors are all sequestered in the matrix. They're not in the cell at all. They're just outside the cell. And these growth factors get released altogether and they activate different signaling pathways. And this, I'm going to try and persuade you, is what's driving repair of the articular cartilage. So I'm just going to talk very briefly about the shear stress, and then I'm focusing mainly on this growth factor repair pathway. So one of the things that we published a couple of years ago is this work that was led by Lini Zhu in my group. And what she was able to show that in addition to the activation of those inflammatory canonical signaling pathways, we also see a drop in retinoic acid. And retinoic acid, or all-trans retinoic acid, is a really important anti-inflammatory mechanism in chondrocytes in particular. So as that drops, you release that anti-inflammatory tone and you amplify that mechanoinflammatory response. So that contributes to this activation of inflammatory genes in response to injury. And what Lini was able to show that if you use a drug that boosts levels of retinoic acid, this is called telaresol, then you can re-establish that anti-inflammatory tone, you can suppress the mechanoinflammatory genes, and in doing so, you disease modify osteoarthritis in the mice. And that was all published. And I say this because it's important just to note that we have an active study at the moment in which we're looking at this drug called telaresol. This is an experimental medicine study. We're taking patients who have hand osteoarthritis. These are patients who are on the routine surgical waiting list to have removal of the trapezium, which is this bone at the base of the thumb. And this is the sort of culprits where the osteoarthritis is. And these are all routine surgical patients. 34 of them we're hoping to recruit. We're about halfway through recruitment at the moment, and they'll be randomized into receiving either telaresol for 2 weeks or placebo for 2 weeks just prior to their surgical date. And at the time of surgery, we take the tissue, we'll extract the RNA from the articular cartilage, sequence it, and we'll look for evidence of suppression of those mechanoinflammatory genes. And if that's successful, then I think that's substantially de-risked a clinical study, so we would apply for funding to do a proper full clinical trial. Okay, so that's the mechanoinflammatory side, and I really want to now come back to these growth factors. So this is where they reside. They reside in this region, which is really quite distinct. It's the pericellular matrix. It surrounds every single chondrocyte within the tissue, and it's characterized by being rich in the heparan sulfate proteoglycan called perlecan. So you can see, hopefully, these little rings of perlecan, little halos surrounding each and every chondrocyte within the tissue. This is human articular cartilage. And you can also see that these two exemplar growth factors, so FGF2 was the first one that I discovered that's released from cartilage. And you can see that both FGF2 and the second one, connective tissue growth factor, which is a member of the CCN family, CCN2, they also co-localize in these beautiful pericellular halos. And what we were able to show at the time is that when you injure cartilage, these factors get released. You can measure them in an injury condition medium. As you can see here, they're not released from rested cartilage, but they are if you chop that cartilage up, as shown there. They're all heparin bound, because if you put them through a heparin column, you lose them in the flow through, but you can get them back again by eluting them with high concentration sodium chloride. And this turns out to be the mechanism by which they're released in the tissue. As you squeeze your cartilage, you squeeze pure water out of the tissue, you increase the local concentration of sodium in that compressed region, and that's sufficient to displace these growth factors, which are just bound by simple ionic interactions with their matrix. So what we did recently is an experiment that we've been meaning to do for a very long time, which is to go back and say, well, you know, we've identified about 5 of these growth factors. I'm showing 3 of them there. But what else is in that pericellular matrix? There must be plenty of other stuff. And so with Anna Hoyle, who is supporting our proteomic sort of effort in the Kennedy Institute. We went back and did this following experiment. We generated some injury condition medium, we put it through a heparin column, and then we eluted it at different concentrations of salt so that we could separate the really strongly bound factors. So this is about 3 molar. We're taking these factors off the heparin column, or 1.5 molar, or lower than that, which we sort of largely discarded. And then we analysed them by proteomics and said, what else have we got in that pericellular matrix that is released upon cartilage injury? And indeed, we found many other things. So pleasingly, we found other members of the same family. So we found a couple of members of the FGF family. In bold are the ones we knew about. So we found actually 4 out of 6 of the CCN proteins. We found this hepatoma-derived growth factor and 2 related molecules, midkine we'd previously identified, and then whole classes that we weren't even aware of. We found the WNTs and their antagonists. We found the BMPs and their antagonist noggin, and we found several members of the TGF-beta family, including this molecule called activin, which is made up of 2 inhibin chains, which will come up later in my talk. So in other words, we found TGF-beta, WNT, BMP, and FGF family members. And why is that important? It's important because of this paper that was published in Nature only a matter of weeks ago, earlier on in the year. And this is the biggest GWAS in osteoarthritis to date. 2 million people, 500,000 thereabouts of osteoarthritis, 1.5 million of controls. And what this allowed them to do was not only increase the number of variants that they found that were associated with OA, they can link those to specific likely genes, putative genes, but then they can take those genes and put them into pathways and say, what are the most important pathways in osteoarthritis? And I promise I didn't cheat in any way, but this is the main paper in their, main figure in their paper. And this is ranked according to how important these pathways are. And we've got retinoic acid, TGF-beta, BMP, WNT, FGF signaling, absolutely everything that we have just described that is part of that cartilage injury response. And remember, cartilage injury is what the epidemiology is telling us. Okay, so we were really enthusiastic about this. So we knew we were onto something that was really meaningful. So what are these growth factors doing? So this is one of Linny's early experiments, where she took primary chondrocytes. After all, it's the chondrocytes that are receiving these factors. And she just did a simple bulk sequencing 4 hours after stimulation with this injury condition medium containing all of those aforementioned ligands. She saw thousands of genes.
From mice or humans?
No, this is pig, actually. She saw thousands of genes that were regulated in response to the injury condition medium. And we were really struck by this pathway when she did pathway analysis. Epithelial to mesenchymal transition is a known pathway that is associated with tissue injury, and it's the process by which your stem cells get liberated, your repair cells get liberated. So it's actually defined, at least in one place, as the process by which cells leave their matrix attachment to become motile and proliferative. And we thought, goodness, maybe that's what the chondrocytes are doing in the tissue. So the next thing we did was to look and see whether they were changing their morphology, becoming motile and proliferative. So if you focus on this top right-hand panel, these are isolated chondrocytes plated at low density and then stimulated with the injury condition medium over a 4-day period. And you can see that by about 24 hours, these cells start to elongate, they mobilize, they make attachments with their neighbors, and then they proliferate like mad. And by the end of the 4-day period, they've almost completely become confluent, and you can see the control cells on the left really haven't changed at all. And you can quantify this here. You can see that there's a partial response to inhibiting the FGF pathway, but it's not complete. So we think that this cocktail of factors being released in the injury condition medium is driving a repair-like, almost stem cell-like response within our tissue. So if these are really repair cells, then these cells should be able to go back to being chondrocytes. And this is what we tested next. We generated these so-called progenitor cells, and then we put them through a standard chondrogenic assay. We gave them TGF-beta plus this cocktail that is well known within the field, and we were able to show that these cells now upregulate chondrocyte genes, collagen type 2, SOX9, aggrecan, and by 3 weeks, these have made little nodules of articular cartilage that looks just like articular cartilage. So we called these mechanoactivated chondroprogenitor cells, and remember, they're derived from the primary chondrocyte itself. So we really thought it would be nice if we could demonstrate this in the tissue rather than just in vitro. And so Lini did these amazing experiments. She first of all developed a method to do single nuclear sequencing from cartilage. Now we had to do single nuclear sequencing because if you take cells out of the tissue, it takes about 6 hours. So you've already activated your injury response. And so doing single nuclear sequencing allowed her to get a 0 time point, a snap frozen time point right at the beginning of the experiment, or she cut the tissue, cultured it for 4 hours or 48 hours, and then extracted the nuclei. And with Steve's help, then analyze this. This is the UMAP that she generated of all the chondrocyte clusters that were evident in her experiment. And if you look at the density maps of these clusters over time, we saw something very striking. So at time 0, most of the chondrocytes reside in these peripheral clusters, but by 4 hours, almost all of those peripheral clusters have disappeared and all the cells have assumed this new phenotype, which is this cluster 1, which remains pretty stable over the next 48 hours. And when we looked at the cluster 1 genes, we found that they were expressing things that migrating cells often express. So they changed their integrin expression, the LAMC2 expression. YAP1 is strongly regulated in these, and DOT1L. And so if we went back to Lini's injury condition medium stimulation, we can see all of those aforementioned marker genes also being upregulated by the injury condition medium. The DOT1L is down here. So that looked very much as though what we were seeing in the dish was indeed mediated by the release of these pericellular matrix growth factors and was happening in the tissue. So can we actually show that chondrocytes really do generate the repair tissue in vivo? And we did this experiment in collaboration with Cosimo de Barry's group, because they had previously done some studies where they'd done lineage tracing of chondrocytes. And so what you do in this experiment is you take TD tomato mice and which are crossed with the collagen 2 Cre, and you activate the transgene at 2 weeks of age, so that all the cells that are expressing collagen 2 then express this transgene, which is fluorescent, but you can also see it with this counter stain. And this stains pretty much exclusively the articular chondrocytes and some of the growth plate cells, which you can see down here. And then we performed a focal cartilage defect. That's done, it's not an OA model, it's a scratch on the surface of the cartilage. You do that at 10 weeks of age, and then you wait 8 weeks. And in a repairing strain of mouse, you can see repair of the tissue. And this is a representative image, which you can see the repairing tissue. So that's where the injury was. And what you can see very clearly is that the cells within it at least are partly derived from those cells that had been COL2 lineage, in other words, cells that had previously been articular chondrocytes. And if you think, oh yeah, but not all cells are labeled, but then neither are they labeled in the articular cartilage itself. And the percentage of labeling in the cartilage is very similar to the percentage of cells that are labeled within that repair tissue. So we think that this is very strong evidence that actually this is the primary mechanism by which cartilage repairs. And you'll see that there are absolutely none of these positive cells in the synovium or even in that bone marrow, which is where a lot of people think repair cells come from. Okay, so I've tried to persuade you that this injury response activates the chondrocyte to become its own repair cell. And this is normally driving the intrinsic repair response of cartilage. So then of course, you have to say, well, what happens in OA? And if we're right that OA is a disease of continual mechanical activation, then of course we should see a signature that reflects activation of this pathway in human osteoarthritis. And so this brings me on to step-up OA. So many of you in this room will be familiar with step-up OA. This is this large proteomic analysis. I still think it's probably the largest endotyping analysis of its sort, at least so far. And this is looking at synovial fluid proteins within the synovial fluid. Huge effort by a number of people, but particularly the ones I've shown here. It's an international consortium, multiple academic and industry partners. And this was our kind of the principles of Step-Up OA. We knew that we were going to have to do this in very large numbers. So what I'm going to present to you today are our established OA patients. We have some other groups within Step-Up OA. That's 1,361 patients with osteoarthritis. We separated these into a discovery and a replication cohort. We had synovial fluid for every single one of these patients. And for each one, we also had cross-sectional demographic data. So we knew their age, their sex, their BMI, and we also knew their pain level and also their radiographic score. And so for each of the synovial fluid samples, we subjected them to the SomaScan assay, which measures over 7,000 proteins in just 100 microliters of fluid. So we did all sorts of analyses, but the most robust of these was looking at those proteins that associate with disease severity on an X-ray. So this is taking a binary cutoff, patients with early radiographic disease or mild radiographic disease and severe radiographic disease into two groups and saying what are the proteins in the synovial fluid that can predict which group you belong to. So what you can see at a glance is that there are thousands of proteins that were significant in this analysis. Everything on the right side of that volcano plot was positively associated, but there were some negatively associated proteins too. Anything in orange meant that it replicated between the discovery and replication cohorts. So this was very robust. And when we did pathway analysis, we found a number of things, but the most significant of these was epithelial to mesenchymal transition. That was the strongest and most replicable pathway through the discovery, replication, and combined data sets. Now, there were some other pathways in there of interest. Complement comes up, coagulation, angiogenesis. These are all pathways associated in animal studies and in human studies with osteoarthritis. So we think that these are relevant, but look what happens when you start to stratify this group now according to some of those other risk factors. So if you stratify by their obesity status, so you put obese and non-obese, and then you repeat that analysis, you look at all the proteins that associate with disease severity in these two groups, you see that the patterns are largely the same. Many of those protein associations are shared between the two groups. But when you do pathway analysis, now you see something slightly different. That epithelial to mesenchymal transition, the pathway of this repairing activated repair cell cartilage is the only one that is now robust across those two groups. And things like complement and coagulation, they're more common in the obese individuals, or at least they come up significantly rather than non-obese. You can do that by biological sex, exactly the same thing. You can look at males and females, the patterns are pretty similar. But now, when you look at the pathways, again, epithelial to mesenchymal transition is the only common core pathway. You can do that by age. You can look at younger people, less than 65, older people, and you see the same sort of pattern. And again, epithelial to mesenchymal transition comes up. So we would argue that this is the core pathway of osteoarthritis, and it does align with this chronic mechanical injury. But at this stage, we're all scratching our heads because we're thinking, well, hang on a minute. We're saying that repair is activated. It's very active in the joint, but it's more associated if you've got worse disease than less disease. So what's going on here? And we turned to some other studies at this stage because one of the only things that is really robust, I think, and lots of people in the field don't believe this, is that the only way to actually get your osteoarthritic cartilage to repair itself is actually to mechanically offload the joint completely. And that can be achieved through this surgical procedure called joint distraction. you put this rather barbaric looking thing, you insert it onto either side of the joint through the skin, the muscle and the bone, and then you put the joint under tension. You literally pull those two surfaces of the joint apart by about 1 centimetre. The individual walks around with this frame in situ. They feel remarkably little pain. They have this in situ for about 6 weeks, and then it's removed and they're rehabilitated. And you can see this is a cohort study that was published some years ago now from Utrecht. And you can see that in these individuals, they have very marked response. So they get an improvement in their pain and improvement in function. And importantly, this is really sustained out to 2 years in this study, but they actually have 10-year follow-up on this group now published. What was more remarkable is that when they looked in the joint by MRI and they looked to see what the quality of the cartilage was like, they were able to show that the cartilage actually thickens and appears to regenerate as a result of this treatment. So here is one individual. This is looking straight on at the knee joint. The white is the cartilage on either side. You can see that this patient at baseline is bone on bone articulation, but by 1 year there seems to be some cartilage in the joint, and at 2 years you can really make out 2 distinct zones of cartilage in the joint. And that was true across all of the 20 patients in this cohort in terms of mean cartilage thickness and reduction in the bare area. So even when you're bone on bone, you get some coverage of that in response to this treatment. So we thought, well, something is happening when you have a chronically overloaded joint that you then mechanically offload. We need to know what that is. And so we went back to Utrecht and we said, can you set up another cohort for us? Only this time, can you give us the synovial fluid? And so what we obtained was 16 paired samples before and after joint distraction. This is at 0 time and at 6 weeks, the time that the distraction frame is being removed. These patients all had severe established osteoarthritis, and we ran it through the Step-Up OA pipeline, and we were able to show 1,500 proteins significantly change just in 16 patients. And we looked at those proteins that were most strongly going down and the ones that were going up, and you can see that many of those TGF-beta family members, activin A, activin AC, inhibin beta A, BMPs 4, 5, 6, and 7, they all plummet in every patient in response to joint distraction. And at the same time, you start to see the emergence of those chondrogenic genes. You see the switching on of type 2 collagen, of the pericellular matrix proteins, the link protein, and the perlecan. And when you do pathway analysis now, you get extracellular matrix organization. So we finally worked out what was going on, and this is what we think is happening in OA.
So did they do that operation mainly on knees, or what joints?
They can do all sorts. I'm going to delay your question to the end because we're recording this. So we think that actually osteoarthritis is a disease of arrested repair. And that's because what we think is happening is that you've normally got this chondrocyte. It's sitting within its pericellular matrix, minding its own business, usually really pretty quiescent, but it is a mechanosensor within cartilage. And so when it senses an injury, you get release of growth factors, and you get this activation of the mechanoinflammatory response, and this converts the lowly chondrocyte into its own progenitor cell. And these are the mechanoactivated chondroprogenitor cells. And these cells do things that chondrocytes never normally should do. They start to proliferate, they start to migrate. And of course, if they're migrating, they have to degrade their matrix as they go along. Now, in an ordinary acute injury response, once those injury signals have gone back into their matrix binding sites, this should just return back through an anabolic phase to the resting chondrocyte, and that is homeostasis. That is the normal intrinsic repair response of cartilage. But of course, in osteoarthritis, where you have that chronic mechanical stress, you get into this chronic cycle where all your cells are chronically activated, and you get this continual migration, proliferation, and degradation of the matrix, which we would argue are absolutely the hallmarks of osteoarthritic tissue. Okay, so the only thing I haven't mentioned so far in my talk is pain, because pain, of course, is vitally important. I also mentioned that mechanoinflammation was inducing nerve growth factor. So what is the role of pain here? Well, when you look at the schematic like this, you can see that pain is a really protective mechanism, isn't it? It's the signal that tells you to mechanically rest your joint to allow these cells to go back to being homeostatic again. And so if we ignore pain, we do so, I think, at our peril. So is there evidence for that? Well, there really is in the literature. So in the last 3 slides or so, I want to just talk a tiny bit about that pain response in the joint. So we've known for quite a long time. We showed it first in the mouse, and then it was validated subsequently independently of us in humans, that if you neutralize nerve growth factor, you have a very powerful analgesic response in osteoarthritis. This is the first sort of major study to be published. This is these are patients who have knee osteoarthritis. This is the placebo response in gray here, and below is a really good dose-dependent reduction in pain with the anti-NGF antibody tanezumab. And this was an effect size that had never, ever been seen before. I'm not even sure in other diseases, really. If you're thinking about paracetamol or a non-steroidal, it would sort of hover just below that placebo. So this was a huge effect size. This was a mega deal in osteoarthritis. And as a result, unsurprisingly, there were lots of phase 2, phase 3 clinical studies, all showing clinical efficacy, but the FDA turned it down. Why did they turn it down? Because 8% of the individuals taking this drug got rapidly progressive osteoarthritis. So they got much worse structural disease. And I would argue that at least some of that, I mean, there are people who argue that there might be other biological reasons why NGF is interfering with the joint, but at least some of that has got to come from the fact that pain is a protective mechanism. It stops you mechanically loading. So we were able to show all of this in the mouse as well, and we published a number of studies over the years to show this. And I wanted to finish with some of Vicky's beautiful work, because I wouldn't be doing due diligence if I didn't include these stunning studies. So this is part of Vicky's recent project. So she's doing this in collaboration with Liliana, who's in neuroscience. And Liliana generated a mouse some years ago, which is very useful because it has a GFP tag that reports on expression of nerve growth factor within the tissue. And this is a naive mouse joint. You can see it's half a knee joint, actually. You can see the two sides, the condylar surface and the tibial plateau here. So all these sort of strongly stained blue cells are actually the chondrocytes. And the type 2 collagen, which you see staining here, is showing the osteochondral junction, which is the junction between the cartilage and the underlying bone. And in this naive joint, you also see blood vessels. These blood vessels are largely within the bone marrow. They course in and out of the bone marrow. But importantly, you see no nerve growth factor expression at all in the uninjured joint. But look at what we see, or rather what she sees, when she induces osteoarthritis. So these mice are now several weeks after destabilizing the joint, and you can see all this nerve growth factor being expressed. These are in the chondrocytes. This is a very damaged region of the joint. I'm sorry, I don't have the osteochondral junction shown, but the osteochondral junction is now around where my cursor is, because so much of the cartilage above it has been eroded. So these green cells here are a mixture of chondrocytes and cells right at the junction of the osteochondral junction itself. And this is where we also see new blood vessels and new nerves that migrate. They change their direction, start migrating up towards the articular cartilage. And at this stage, the mouse is literally walking on its nerve endings. So it's hardly surprising that this is a painful disease. Here is a huge osteophyte on the side of the joint. I'm not showing you the synovium in these images, but I have Vicky to vouch for the fact that there is no NGF in the synovium at all. It really is almost exclusively in the cartilage and on the cartilaginous cap of the osteophyte at later stages of disease. Okay, so I'm going to finish there by saying that there are a number of things we didn't know, but I would argue after 25 years, goodness, I should have discovered something, shouldn't I? We really do understand them now, and perhaps for the first time just in the past year or so. We do know the molecular pathways that initiate and propagate disease. These are highly mechanosensitive, and so they align with the epidemiology. They also align perfectly with the GWAS hits that have come out in recent years too. So we're very happy that that biology is really relevant. We do know where pain comes from. At least we know one of the major pain drivers, which is this nerve growth factor. And this seems to be also driven by those injured chondrocytes primarily, and that changes the neurovascular structures within the joint, and that perpetuates this chronic and acute pain. I didn't show you all the primary data from Step Up OA, but we know that there are not distinct diseases that all converge on a common pathway. We think that osteoarthritis is a common disease. It has this core pathway, which is this epithelial to mesenchymal transition. This is the signature of that damage activated repair response. And really, for the first time, we're calling this a disease of arrested repair. We mustn't downplay it. I mean, this is chondrocytes and cartilage doing what they should do. They're responding to injury to activate repair. They just get stuck in this chronic activated state, and they need to return by mechanical offloading. And the big question for us now, of course, is whether or not we can do that pharmacologically in order to switch them back to their original phenotype. So with that, I left, I hope, a decent amount of time for questions, but I want to acknowledge my team. Glad to see some of you are here today. That's good. And our wider OA centre. We set this centre up 13 years ago, and we still meet every single week. And we share data, we share ideas, we share journal club, and we still meet. And occasionally, this is post-pandemic, this trip to London we had. So we're multi-institutional now, which is why we only meet online. But we're still a very strong and vibrant community. And we had our Cutting Edge OA meeting last week in Oxford, which was fabulous. And we have people not just from the UK actually, but from Berlin. They came over for it. So it's a really thriving community. And obviously to my funders and to you for listening.